speaking of Mercury, that's what our topic is going to be tonight, and I'd like to hand out, there was a Mercury forum that was done about a year or so ago, and we put together a little fact sheet on Mercury. And I'm going to hand these out so you've got some of the basic information on Mercury, but really what you're going to need this for is on the back so you can take notes. I heard Joel talk just a couple days ago, and you're going to have a lot of information, good information, and you might want to take some good notes. So I'll pass this around so you have that. And I'll introduce our speaker here. Joel is a member of our chapter here tonight, but he's got his EPA hat primarily on tonight. He's going to be talking about data that the EPA has collected, the ECA and the DNR in the St. Louis River estuary. And he's also going to not only just talk about the science of how mercury gets into the food chain and how that all works, but also a little bit about how that affects things like the area of concern, the total maximum daily load plan that we'd like to see done on Mercury for the St. Louis River. So he's going to try to tie all that together for us. Joel, since 2009, he's been a research biologist here at the U.S. EPA lab. And it's really called the Office of Research and Development, National Health and Environmental Research Laboratory, Mid-Continent Ecological Division, Ecosystem Assessment Branch in Duluth. That's why we all call it the EPA lab. I didn't realize it was that complicated of a place. And Joel is also an adjunct professor at the UND biology Department of Biology, I should say. And Joel did his undergraduate work at the University of Ann Arbor, Michigan, and he has a PhD in marine science from the College of William and Mary in 2006. And his interest in research is in how human impacts the environment influence aquatic ecosystems and fish populations, especially with a focus on aquatic food web dynamics, fish movements, and fish habitat. And he uses eco-geochemical markers, which maybe we'll learn a little bit about tonight, and hydroacoustics to study fish. And for a researcher who's relatively early in his career, he's got an impressive list of publications and awards and so forth. So please join me in welcoming Joel Hoffman. And he'll be speaking about getting to the body of Mercury and the St. Louis River.
Some of you may have found a subtle pun in that title. For those who haven't found it yet, I'll let you dwell on that. Is there a remote for this? Or just...
No, you want me to run it for you?
Sure. Yeah,
okay. Maybe is that?
If you can put the second one back on, I think that's better, because then we can see Joel and his
You're almost asleep.
This one?
Yeah,
Okay.
because then you can see Joel.
That work?
All right.
Rich, I got a chair for you up here.
Okay.
All right. So what did you... Two big things tonight. One is I do want to introduce you to some of the ideas behind what influences mercury in fish. And this is a really tough topic. It's complex because there's an environmental component, there's a human component, there's a geochemical component, which confuses all of us. And so I want to walk through that in a careful way and kind of help give you a better understanding of what's at stake in terms of how this contaminant works in the environment, because it's tough. It's a tough issue. The second thing I want to do is actually show you some data. I'm going to show you the real data that we in PC are collecting. And I do that not just in sort of a sort of good faith sort of way to have some transparency, but I also want you to understand what the biologists are looking at as we work through this tough topic. Because it is tough. And I think it would help all of you to understand that there is not to sort of break through the mysticism of data and see what we're actually collecting out there. And they're not complex slides, we'll walk through them. But I do want you to see them and understand them. So, next slide, Rich. We're going to do this as a story in 3 parts. First thing I want to talk about is return of a river. I'm going to talk to you about how the St. Louis River has recovered from nearly a century of degradation. Show you some data and explain why that's important for the conversation about mercury. I want to give you a sense of perspective and talk about mercury as a global environmental problem. Because we can't understand what's happening here until we really understand the regional, continental, and even global scale of mercury as a pollutant. Because all of that comes down to explain why we have a problem here in northeast Minnesota. And the last part of my story tonight is going to be on mercury and St. Louis River fishes. We're going to share some data talk about what it means, both in terms of the science and in terms of human health. So return of a river. When we first settled this system, it was a vast wetland. I think many of you in this room know this story. I'm not going to dwell long on it. It wasn't long before, after early settlement, population began growing rapidly through the 1880s and 1890s. And we know this story. We know that as part of that settlement, one of the first things they did was denude the landscape and radically altered the basin and the hydrology of it through early timber industry. I'll show you some photos of that. As the industry developed, we'll hang there for a second.
Gotta go back.
Yeah. And as the industry developed, we would start to carve out large pieces of wetland from the lower harbor. We'd establish ports and industry. And all these had a long-term cumulative impact on the river. But we're back from that now, and I want to walk through a few data pieces on that to explain why that's relevant. So, just a reminder and orient ourselves, the St. Louis River is truly a national resource. I mean, it's an impressive system. It's the second largest tributary to Lake Superior, the largest in the U.S. It's quite long, almost 200 miles. Its watershed is massive, over 3,600 square miles. And within its lower reaches, it contains the largest freshwater estuary in the Great Lakes, a place of tremendous ecological importance. Over 12,000 acres, extremely high biodiversity in terms of the fish and the wildlife. It has a very important role in the continental scale ecosystem through its importance for migratory birds and wildlife. Extremely high abundance of coastal wetlands, which makes it very highly productive. And it's marked, and this will come into play later, by a unique property. It has bidirectional river flow multiple times a day, owing to a seiche, the forcing of waters into the river, owing to complex lake wind-driven forces on the surface of Lake Superior. And we'll get to why that's an interesting feature in terms of the Mercury discussion. Maybe you've seen some of these photos or photos like these that describe how we change this river. And of course, here's two most glaring examples. One was not just the timber industry denuding the landscape and taking the trees off, but increased sediment deposition to the river, made the river flashier, moved more sediment material into the river. But of course, the history of the sawmills was leaving a vast amount of material in our riverbeds. And much of that remains. And much of the restoration work being done is about removing a lot of that wood waste from sawmills and other facilities. And of course, we had the rise of a lot of heavy industry. The one that perhaps had the greatest environmental footprint on the system was the U.S. Steel plant. And we, of course, have that legacy today. We now know is the U.S. Steel Superfund site. But it was operated, of course, for quite a long time and had a tremendous environmental footprint and a tremendous economic footprint on the region. The impacts to the system were severe, and they happened actually relatively rapidly. So this is from the Minnesota State Board of Health report in the late 20s. I'll read it out loud, so I suspect on the back it's hard to read. Quote, near the upper end of Spirit Lake and McDouble shipyards below Spirit Lake, samples showed that the bottom was covered with a thick layer of mud, thoroughly impregnated with small globules of tarry substance. The mud and water in this portion of the lake had a strong creosote odor, and the oily film covered the surface. No living organisms were found in the mud sample, and it is improbable that even adult fish could enter this zone with impunity. We're basically talking about the river between Cloquet and Spirit Lake being essentially biologically dead by the 1920s. There were early reports. The USGS actually did a survey of the river in 1905, and they didn't have a lot of biological data, mostly chemical. But if you read into the alkalinity data and some other pieces, it suggests that the worst probably happened somewhere between 1905 and 1928. But in 1905, there were clearly impacts. There was awareness enough that we had federal surveys on the river. But it appears, at least from that data set, that perhaps this somewhere between 1905 and the mid-20s is when this truly set up to be this severe. And it continued. This is from the Minnesota Division Game of Fish Survey of the River, 1944. Again, I'll read, quote, from the fisheries point of view, the greatest pollution problem in this river basin is the discharge of municipal sewage, paper mill waste, wash water, and other industrial waste at Cloquet. Both the 1928 and 29 survey, one previously referenced, and the present survey show that the effect of this pollution extends to St. Louis Bay and has nearly eliminated fishing and fish life from this lower stretch of the river. Other sources of pollution in the lower river are sewage from the city of Carlton, waste from steel mills, shipyards, and other industrial activities near or on the estuary. This was a long-term period where we had persistent episodic anoxia. That's the condition in which there is no oxygen in the water so that no animals can breathe. It simply is a block of all aquatic life. They cannot persist in those conditions. And on top of that, we had the addition of a tremendous volume of organic pollutants, things like pulp waste, organochlorines, dioxins that were coming from industrial activities, and metals from the heavy industry as well. And this was all going right into the river. This is a map from that 48-49 survey. I know you can't read it. There's some pictures to highlight a few features. I do want to point out that every place where there is text written on this was an industrial site known at the time to have a problem associated with it. It's worth going back and looking at some of those maps if you haven't done that. There's actually a number of copies that's reported around the region. If you were looking for a copy, I have one. It's free to borrow. It's quite a read. I want to talk about three features of this, if you want to just hit the go button there, Rich. One is some of the worst sites were, of course, eventually flagged as Superfund sites under the Superfund program. Of course, I think most of you are aware, the Interlake site was cleaned up. It was capped, part of it was capped, part of it was dredged. It's in a recovery phase, under monitoring. This was a significant and highly polluted site. In many ways, however, compared to the job left undone, which is the U.S. Steel site, which is a site that is so large, in many ways it boggles my own mind. It is a massive site because it includes, here's the original U.S. Steel site, includes both a large contaminated area here to the north, this large bluff area, which is also all contaminated material, and also more to the south, down at the bottom of Spirit Lake, which is off at the bottom of that map. So it's a truly massive site. And of course, the assessment of that site is ongoing. Hope for remediation planning coming in the near future. That's a tough one. The other remarkable thing we'd like to talk about, Rich just made a mention to, was that at the time we had these unconsolidated sewage systems. Local neighborhoods had their own systems. Primary treatment was possible. That would basically be a pond. Secondary treatment, some filtration was happening, but the system was highly prone to overflowing. It was very unstable. And so by and large, a large amount of sewage material was making its way to the river, by every account we have of the way the system functioned. And so the creation of the WSSD was important for two reasons. Both because it eliminated that problem, in the sense it consolidated those and created real treatment in an organized fashion. The other thing, of course, it did is, and I think this is a really remarkable insight they had, it was to include a lot of the industrial facilities. Make sure the industrial facilities, rather than having their own discharge systems, were sending their water to the treatment plant. That's actually fairly innovative. Few systems in the country are set up like that. And it's a smart thing to do. So that was another nice piece of OSSD. What's wonderful at this time is that the MPCA was collecting data through the Milestone Monitoring Program. They actually have routine monthly monitoring located here, and they had other areas too, but these are particularly useful data sets. One here in the upper estuary at the Oliver Bridge crossing, and one down below at the 53 Blatnick Bridge crossing. Why are those useful? They're useful because they're constrictions in the river. So I can take a single sample there and get a really nice representation of what the quality of that water is coming through there. And they were consistent monthly data. So it's a very nice time series on this river. So I wanted to talk to you, show you a bit of that data, and we'll forward. This is a plot over time, 1950 to 2020, of the dissolved oxygen levels in this river. That's DO, dissolved oxygen. And so, in 0, 2, 4, 6, 8, 10 milligrams per liter. We generally consider 5 to be potentially limiting to aquatic life. Anything below 2 becomes extremely threatening. And so, you can see back here in the 1950s and 60s, these are routine, extreme hypoxic events. Nearly no oxygen in the water up at Oliver Bridge. So this is right in between this area, peak pollution, right? The pollution ran from Cloquet somewhere down to here, we think, based on the historical descriptions. This is the area that was essentially biologically dead. And so this is right in the heart of it, and it's represented well in the data. These are summer low time. July through August averages. That's typically when we see the lowest dissolved oxygen in the river. There's biological reasons that's true, and so remarkably, somewhere around 1970, things start to get better. By the early 70s, we actually come out of that period of periodic hypoxia. And the last time we see DO levels that are potentially parented to aquatic life are somewhere in the late 70s. I believe the year is 1978, the best of able to piece it together from the data sets. And since then, we've had continually good oxygen in this river.
What's going on since 2010?
That's a really interesting phenomenon. It drops back down. And we now see this persistently, that there is actually, we get sort of 7 to 9 milligrams per liter, which suggests that there's actually more respiration in the system than production. In other words, there's more bacterial activity than carbon being fixed by algae. If you think about this system, however, it's really a vast wetland. It should have a really strong metabolic, it should have a lot of microbes doing a lot of things because we have a lot of shallow water with a lot of sediment. And that's perfect habitat for microbes. So in some sense, I'm not sure this is unhealthy. We thought about this a lot. And I would actually say this is probably a good thing. The best way we can interpret this is it's a signal to the point where the wetlands start to become functional again and do what wetlands do. And one of the things they do is break down organic matter. And in doing so, they consume oxygen. So these levels are never at a point where they impair life. And it's very typical in productive wetlands to see a signal like that. But we do continue, since that time, even our more recent data, we continue to see summer levels that are in that range. Yeah. This is a little complicated. These are two key parameters to understand when we talk about this debate. So let me walk through these. Again, these are time series. White is our upper estuary station. Here's the Blatnick Bridge station, that's in the dark. And here I put on a log scale the total phosphorus, which is a key nutrient that can fuel algal problems in the system. And historically, we knew that there might have been algal problems. And I'm going to focus just on this plot for a second. But what you notice over time is that the amount of those nutrients coming down the river has gone on a very steady decline. That's a great sign. This system is coming back to being healthy. We had too many nutrients. And in fact, this green line represents, for the area of concern, for the management plan, the target goal for nutrient levels in that river. And we're coming in line with those today. It's actually really exciting. Where were these nutrients coming from? Well, in part they're coming from us. They were coming from our sewage system that was malfunctioning and sending human waste and other organic material into the river. In part came from the other types of pollutant. But a lot of it also came from sediment. This was the denuded landscape where that water, that phosphorus is tied up in our clay soils. And every time it would rain and those sediments would wash in the river, the phosphorus would come with it. And as the landscape has recovered, you see this all over the Great Lakes, as the landscape's recovered, total phosphorus has dropped. As trees have come back and forests are being managed in a much more sustainable manner. And so you can see this consistent drop at both stations across the estuary. That's fantastic. Great news. However, we also know that mercury bioaccumulation actually increases as ecosystem productivity decreases. Let me say it again. Mercury bioaccumulation of fish actually goes up in systems that are less productive. And this becomes systems becoming less productive because phosphorus is going down. We see that all over in lake data. Lower productivity in the lake, the fish have more mercury. This is the total suspended solids. So this is a measure of sediment coming down the river. Again, a time series. It's in a log scale. We do that because it makes these plots much easier to read. So interpret more with these patterns. The lines show you the patterns. We see it going down as well. Total suspended solids, minus the 2012 flood, are going down routinely in this river. It's another good sign the watershed is returning to health. It's going down a little faster at the lower estuary and then in the upper estuary, which is interesting. You can think of the difference as kind of a pollutant signal. What's really happening here is that because we stopped pumping a lot of suspended solids vis-a-vis a malfunctioning water treatment system, discharge of other pollutants, which were in solid form, and we're seeing a little faster recovery here. So this is great to see. It's declining still. And the good news there, we know across Minnesota, a good predictor of mercury in the water is actually the total suspended solid. The mercury falls onto the landscape and moves with the soils back into the river when it rains. And mercury delivery therefore decreases as TSS concentration declines. So that's a good piece of the puzzle. Rich wasn't kidding about the return of the fish. This is one of the most biodiverse habitats in the Great Lakes. This is my personal running list of fish that we have caught out there. Actually, minus one. One of these is a John Lindgren fish. I just poached it because it's awesome. They were out fishing a couple years ago and caught an American eel in their nets, which is, I just think, remarkable. We haven't caught one yet, but I borrowed it, John, sorry. But over 50 species of fish. Over 50 species, just in the St. Louis River estuary. Now, everyone with an asterisk is a non-native fish, but nonetheless, so we had some artificially augmented biodiversity in this system for sure. Nonetheless, we have tremendous biodiversity in this system. And of course, I think sort of what's become sort of the icon of the recovery of the fish has been the recovery of lakes. It's really a wonderful story. So against this background of recovering river, it's truly frustrating that we can't eat the fish in it.
Question, Joel,
Yeah.
is with the increase in exotics, apparently we haven't lost any species yet as far as...
No evidence that a fish has been extirpated. So just as a sidebar here, I won't dwell too long on this. So the biggest concern certainly has been over two species of concern, round goby and ruff. And based on our work with Fish and Wildlife Service, those species are actually in dramatic decline right now. And we're not sure why. But the ruff are actually the lowest since their first detection in the river right now. They're actually hard to find. It's really interesting. And some of the native species that they compete with are doing quite well. We had incredible catches of trout perch last year in the nets.
How about smell?
Smell are down, and they'll probably, as long as lake trout are doing well, we will have fewer smell. Yeah. All right. The river's healthy again. So let's come back to the Mercury story. Mercury is a global environmental problem, and it begins at the top of this plot. This is sort of a generic schematic of it, and if it looks complicated, it is. And that's one of the problems with this conversation, is that this is really complicated. There are lots of processes running lots of different ways, and so there's lots of different things that can mean that the fish that you would like to eat is a threat to your own health. But it begins here with emission and transport, long-range emission and transport. I'll show you a slide to figure, but that's a continental phenomenon, and even an intercontinental phenomenon. So we can have mercury burned in Asia showing up in fish in the St. Louis River. So it begins here with our connection to the rest of the world through atmospheric transport. That material comes down onto the surface of our land and waters as both wet and dry deposition. It can transport into rivers as mercury, inorganic mercury, or slightly charged mercury, and become reactive in the food web. It can also land in wetlands, and wetlands are really good at what's called methylation. That's the process of taking inorganic mercury, and bacteria, sulfide-producing bacteria, will actually attach to it a methyl group, and in this quasi-organic form, it becomes readily available to the food web, and it bioaccumulates. Once it becomes methyl mercury, this is the toxic form. Animals cannot simply pass this through their kidney or intestine systems. It bioaccumulates in the tissues and it's a neurotoxin in this form. And so it passes through the food web from algae to the lower food web to fish and then to people. And in the meantime, the other mercury is cycling around and around in lots of complex ways. Through the sediment, back to the water, and that's what makes it really, really hard. But what I want to do, rather than getting into this diagram in detail, is talk about some of the features we know are important and how they relate to the river. So here's what we're worried about. Simply put, methylmercury is the most toxic form of mercury in that methylated, that semi-organic form, and as such it serves as a neurotoxin. So it impacts the immune system, the nervous system, and of course we know that developing embryos and children are at higher risk than adults. For the Great Lakes, the estimated economic cost of decreased productivity owing to methylmercury exposure may be as high as $435 million annually. But it's a really hard number to get at. Extremely difficult to estimate. Exposure to methylmercury in the U.S., based on FAA reports, is primarily through the diet. The vast majority of methylmercury Americans get is by eating fish and seafood. Nationwide, actually only about 15% is from freshwater fish. I suspect it's much higher locally. I've come across good local numbers for that. I think most of us would prefer to eat walleye than tuna. I would. Exposure is a concern locally. Rich just handed out that data sheet. This, I think, really became to light recently. Pat McCann at the Minnesota Department of Health was the lead on a very nice study surveying, looking at cord blood samples across the basin, and a surprising number of cord blood samples from the region came up high in mercury, suggesting that the levels, about 10% of the women had levels that would put their developing fetus at risk. That's not good. The levels of concern, if you can kind of hold on to these numbers in your brain for a little bit, it's about 0.2 parts per million. Parts per million. 0.2 of a part per million. Very small amounts for adults and extremely small amounts for children and pregnant women. The maximum consumption we allow for adults is almost 3 ppm. We say do not eat if it's about 1 ppm for children or women who are either pregnant or might be pregnant. And that is the Minnesota Department of Health standard. Okay. So where is it coming from? About 30%, this is from the PCA, about 30% are natural emissions, 30% come globally from all over the world, 40% are from greater parts of North America, 10% comes from Minnesota itself. Of that, 10%, most of it comes from burning fossil fuels, and then it's evenly split more or less between the product use and misuse, disposal, and then the attack and net processing, which of course is a local source for us. So, yep, that's perfect.
Okay.
So let's look at what that really means. This is a plot, this is a map of mercury emissions. Don't get too hung up on the scale. The hotter color that is purple, red, those are areas of higher emissions. And you can see that in general in the U.S., emissions are higher in big urban areas, and we have this classic phenomenon where from between Minnesota down through the upper Great Lakes, lower Great Lakes region, and out to Maine is kind of an area that we have traditionally had a lot of problems with mercury in the U.S. And you can see that emissions are high, and those with really good eyes will notice that the St. Louis River watershed area is among one of the purple patches. So locally we have moderately high emission of mercury, inorganic mercury into the environment to contend with. This is the flip side of that. There's what we put in and there's what falls. This is the map of the wet deposition. Red again, high deposition, yellow, medium, and then light green gets into lesser deposition. Notice that there's some deposition nearly everywhere. We are in this sort of low middling range up here in our part of the region. So it gives you a sense of sort of where we fall in terms of our what we are adding to our local rivers and environment. This is actually what's in our water. This is from a report from Bruce Munson. Bruce and I have been collaborating on some of these data sets for a few years now. This is on the left, this is the total mercury. So it's the total inorganic, which is often there's a lot more inorganic than methyl. And so it's not great, but it's the methyl that's really a problem. And so we also take measures of the methyl. For those who have good eyes will notice that here is the St. Louis River watershed. For total mercury, we are on the high side. We rank with the highest, those are the red, the hot ones, the upper quartile. The upper 25% of watersheds measured in the state of Minnesota. So we have high mercury in our waters. We're in the upper quartile. We're ranking with the other high body waters. The methyl side is maybe a little better. We are actually in the third quartile, so in the 50 to 75th% range. We are not the highest for methyl. Some of those are in the south and in the northwest corner. And then, I alluded to this already, I just want to show you the data we're working with. This is again from Bruce's work. They looked at sort of the correlates for what was driving these levels of mercury. And I wanted to point this out because it's clear that the St. Louis River has high levels of total mercury. So this is total mercury plotted as a function of the total suspended solids, which we talked about has been going down steadily in the St. Louis River. What you can see, is what I saw earlier, is that there is a relationship where the more suspended solids you have in your river, the higher the total mercury. So this is why it's a good thing that we're walking back this way. This is the best predictor of mercury in Minnesota streams that they could find. If you know what the suspended solids load is, you have a pretty good idea of what the mercury would be. And so PCA, Bruce Munson's report, probably the best thing we can do in the state is if we get suspended solids down, we can impact mercury. So what influences mercury in food web? So that's sort of the what comes in, right? That's the contaminant as it enters the environment. But then it still has to get up into the food web, into the fish, which is the thing we're interacting with. And broadly, I don't get into the details of this for too long, but I want to point there are three big ways to think about this. First of all, it's mercury bioavailability. What I mean by bioavailability is kind of a funny jargon one. What I mean is mercury can actually get into the food web. So it basically has that methyl group attached. It gets methylated. And so mercury bioavailability depends on, of course, how much methyl mercury there is, but it also involves a lot of complex chemistry. When you have organic bound to a metal, there's an opportunity for lots of complex chemistry to occur. And so there's lots of other kinds of chemical interactions in the water that can drive that to be more or higher or lower in concentration. There's also the bacterial activity. Because that methyl group's attached to the mercury by bacteria, how active the bacteria are make a big difference. And things that drive bacterial activities, things like temperature and the kind of bacteria that are there, can also influence how much mercury gets up there. So those are all those are factors. And then to get into the food web, then we have to deal with a whole other suite of factors. Things like what kind of ecosystem is it? What's in your local land cover? Where does the fish feed on the food web? How old is the fish? All those factor in. So it's very complex. It's a tough topic to talk about because when we talk about, someone says, well, the mercury is high for this reason, the scientist looks in their back pocket and says, well, I got a list for you. It could be this, or 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 this. And we have to slowly work through it to understand what's going on. And it takes some time. But here's the problem, and it's a remarkable problem. Few contaminants behave this way. So let me walk through this for a second. This is mercury in parts per million. So that's 1 part per million. That's 1 part per trillion. That's less than 1 part per trillion. These box plots represent typical ranges or values you would get for things in the water, as methyl, in the water, the algae, the zooplankton, the little grazers that eat the algae, the fish that eat the zooplankton, the fish that eat the other fish, and then in this case, loons, birds that eat those fish. And there's 2 pieces of this plot. This shows you the amount of mercury, and this shows you how much of that is that toxic form, the methyl, and that's percent. So let me walk you through this. Notice that in natural waters, mercury is often on the order of 1 part per trillion, and the methyl part is a minor fraction of that, less than a part per trillion. And here's the remarkable piece. Just in that first step, phytoplankton are incredibly efficient at scavenging that methylmercury from the water column and incorporating it into their cells. And so there is a remarkable jump here, 4 to 5 magnitudes of order in concentration to accumulate that mercury into those cells. It's really amazing when you think about what they're doing. And so here's where the problem begins. And then when it gets into the food web, up from there, it jumps another order to 2 in magnitude and concentration. This is the biomagnification effect. An order of magnitude increase in concentration from something that's 1 in 10 to 1. Something that's 1 in 1,000 to 1. It's incredible. And so this is what we're facing, is by the time we get into piscivorous fish, the walleye that we want to eat, we now have levels approaching 1 part per million, even though we started at the beginning at less than 1 part per trillion. And here's what happens to the methylmercury. At the bottom of the food web, it's a very small proportion. There's not much of this neurotoxin up there. But as it gets up into the food web, it rapidly becomes a dominant form, because that's what's bioaccumulating. These animals can get rid of inorganic mercury. Their body can expel that, but it cannot expel this methylated form. So by the time you get into these upper trophic levels, nearly 100% of the mercury in your body is this methyl form. So that's what we're dealing with. This is a schematic that I think is helpful. I'm going to point out a couple of features. This is describing some of the key features that really influence mercury in the environment. And I want to start up in the top of the watershed and work our way down. And so when you imagine this being the upper watershed, this being the fringing zone around the river, and imagine in your mind that this is the St. Louis River and its estuary. We start at the top, and one of the things that happens is in forests, we get more efficient deposition, wet and dry deposition, onto the soils in forests. It's just through the litter fall. Litter fall actually enhances transport of mercury onto the soil surface. So if you have a lot of forest in your watershed, it actually enhances the transfer of mercury into the environment. That material washes down and through the soils, At the fringing part of our watersheds, we have these fringing wetlands. These wetlands are sinks for mercury, it accumulates there, and they're really good at methylating it. So you have lots of wetlands, there tends to be higher mercury levels in your fish. Especially if those wetlands have water flow running through them, where that water carries through the groundwater that methylmercury out into the local waterway. And so these shallow water flow paths can contribute a lot of methylmercury by driving the mercury out of that wetland and into the local waterway. So the local hydrology is really important. In our case, we do have this feature, right? Our geology really favors these shallow groundwater flows. So we're probably moving, we're set up to move mercury from these wetlands out into the river. And of course, once it gets in here, it gets into the fish. One of the other features that can really drive this are periodic, long-term water level fluctuations in and around these wetlands. And it's the wetting and drying of that wetland surface that can actually enhance methylation. And this has not been studied well in Great Lakes coastal wetlands like the St. Louis River. But one of the things that system is subject to is long-term changes in lake stand, where marshes can get stranded and then flooded over years or decades. But it has not been studied to what effect those long-term changes in lake stand can impact methylation. But everything we know about from lakes and reservoirs would suggest that that could be an important factor to consider. The other piece I talked about is this part, the low productivity. So in ecosystems that are relatively low in productivity, we have higher bioaccumulation. So here's sort of things to think about then. In the St. Louis River, we have moderately high wet and dry deposition. We looked at that in the deposition plots. We have a highly forested watershed. We have extensive wetlands in the upper and lower watershed and extensive fringing wetlands on the St. Louis River estuary. We have changing water levels, both in our reservoirs and in the estuary itself, owing to its connection to Lake Superior. We have declining and now increasingly low total phosphorus to the system. And the other piece I didn't talk about much was dissolved organic carbon. Dissolved organic carbon is small molecules of carbon, obviously dissolved in the water. You get them a lot where you have a lot of wetlands. It gives that water that dark tea stain that you see in the St. Louis River. And that is really an important piece of the puzzle because it's that carbon that the mercury transports itself on into the river. So you have lots of DOC, lots of dissolved organic carbon. We also tend to have a lot of mercury. And the St. Louis River has that nice dark tea stain because of its association with a lot of nice wetlands in the upper watershed. And that becomes a delivery mechanism for mercury. Go ahead. So, as we talk about the river, let's talk about the elephant in the room. And started, I think everybody has their own story about how this started. I'm going to give you my version of it. And I think it began from Josephine's reporting on this issue back in April. And this was, of course, the TMDL was moving, people were aware of it. There had been some commitment on the MPCA and the partners to start moving on it. And then all of a sudden, somehow the St. Louis River and some other watersheds were off the list. And so this was the quote from the Star Tribune article from Josephine Mercati. Officials from the Minnesota Pollution Control Agency said they are committed to reducing mercury pollution in the river and the 10% of the state's waters that have unusually high levels. But, they said, the agency's lead scientists believe the state first needs more research on how mercury behaves in nature and why mercury levels in fish from the St. Louis River are significantly higher than those elsewhere. And all of a sudden, in this article, the St. Louis River is put on the map. And maybe many in this room know more than me than how that came about. Of course, there was a quick reply from the MPCA Commissioner, John Lake Stein, who had a broadly published editorial on the issue, who said that our decision about the St. Louis River Mercury Project focuses on the 10% of waters where the mercury problem will not be fully solved by the 93% reduction plan. That's the plan the state's proposing to get its contribution of mercury down. And these waters, which include the St. Louis River, mercury is more readily available to the food chain. This results in higher amounts of mercury in the fish. To further reduce mercury in the fish in these waters, we need to understand what makes the mercury more available and how to reverse that effect. And so this is why this sort of became news, right? Because we have a lot of people invested in time and energy in getting mercury out of the environment, and there's a perception that they're backing away, and the perception seems to be based on the St. Louis River. it's a little more complex than that, and I think we need to be careful here. There are actually two different actions of relevance to this conversation. Let's not lose track of that. One is the Great Lakes Water Quality Agreement. The St. Louis River is called the Areas of Concern. This was designated long ago under the original Great Lakes Water Quality Agreement in the 80s. Let's be clear about what that is. That identifies that we have a binational commitment to get the St. Louis River cleaned up of its legacy sediments, a binational commitment. And the EPA is committed to supporting the states in those actions. The Great Lakes Legacy Act was written to provide funding to get those cleaned up. It's been enhanced by the Great Lakes Restoration Initiative. That's all good news. But a couple notes here. First of all, the Great Lakes Water Quality Agreement is just that. It is not regulatory. And actions associated with it are not enforceable. This is not a matter of law. This is a matter of agreement. Of course, the focus is on source of legacy, sediment contamination, and a major priority right now is getting these cleaned up. That contrasts in many ways with the TMDL process, which many of you understand is an acronym, but that's actually part of the Clean Water Act 303D list of impaired water activity. And so once your impairment, you have to act on it. That's what the Clean Water Act says. It's regulatory activity. It is enforceable. So it's a whole different thing in terms of why people act. The focus is on the source of impairment for TMDL. So even though you're trying to reduce a load, that's the L, you're trying to reduce the amount of contaminant you put into the environment, it has to respond to the impairment. And so in this case, the impairment is the fish tissue. So there has to be a connection there. You have to be able to make it. Which makes this a little complicated sometimes. When we have a biological response and an inorganic, like a metal, contaminant. And of course, we express a lot of the activities, not exclusively, but we've been discussing it certainly in this case, through the imposition of a total maximum daily load. In other words, we're going to limit down the amount of pollutants that go into the waters every day. So let's come back to the elephant in the room. You know, do St. Louis River fish have a nuclear high mercury? Is that what we're really talking about? Is that even the right question? And I think that's where the science can contribute. We can at least talk about what is the real levels of mercury in these fish, and let's have an honest discussion about where we are at with that. So that's what I want to do. I want to show you how much mercury is in St. Louis River fish, and how it compares to other state waters. What are our local sources? We have some. We have taconite mines, we have coal-burning power plants in our watershed. We have coal-burning plants just outside our watershed that contribute to our watershed, possibly. We have wastewater treatment. We have legacy sediment contamination. We have mercury in the sediments in the estuary. And we have some natural vulnerabilities. We have mostly forested land. We are a wetland-rich ecosystem. We have high inputs of dissolved organic carbon, this carrier for mercury in the environment. We have reservoir systems, which tend to also have higher mercury because of changing water levels. And we're in northern latitude. And it turns out that when fish grow slowly in cold waters, they bioaccumulate more mercury. So we have some natural vulnerabilities. We're a sensitive place. Mercury and St. Louis River sediment. So this is the public database for sediment contamination in the data. This is the surficial sediment data that we've been able to plot and collect. I just want, don't work on the values here. This is the amount of inorganic total mercury out in the sediment of the St. Louis River. And I do want to point out we do have some issues. Those are in red. Those are our hot spots. And the one that's most visible, there's U.S. Steel. So it is true, we do have a sediment problem still. And there are clearly areas around here, around Barker's Island, some of this looks back in here, the data points are covered, around WSSD, where it's been recognized by the partners working on this issue that there's cleanup work to be done, and that includes dealing with mercury. So we do have mercury in our sediment. There is monitoring of mercury in St. Louis River fish. Let's be very clear about that. Both the Minnesota DNR, in conjunction with MPCA, and the Wisconsin DNR, routinely, though not annually, conduct monitoring. But we have some scientific caveats. It has a limited spatial emphasis, so we can't talk about the whole watershed everywhere with all the different impacts. Not historically, not using the data that's out there. The other is traditionally they've limited to a few number of species, so it's really hard to we have an apples and oranges problem often where we're not getting lots of the same species everywhere, and there's only a few of them, which gives us a narrow window into the environment. And that's tough when you have a complex environmental problem like this. So we do have some limits about what we can do with the data we have in hand. With that in mind, Bruce Munson, MPCA, and myself got involved in 2013 in trying to do a much better job of characterizing the entire watershed. So we basically broke it up. He had a mandate to work above Fond du Lac Dam. He cooperated. It was DNR and PCA and Fond du Lac Natural Resources all got together, got people and sampling equipment, and went out and caught fish. And our team at EPA worked with them, with Minnesota DNR and PCA, working below in the estuary. So we were hitting, but you have to appreciate the effort here. We were hitting a huge range of habitats: streams, reservoirs, main stem river, and the estuary. So we have to cover our ground, and we ended up with a very diverse group of species, 13 species in all. So that was great, and I think a big first step in this toward making progress. So what I think of this exercise as, when I look at this diverse range of habitats and diverse group of species, I think of it as a range-finding exercise. What we can tell you is exactly where all that mercury is coming from in the way we collected this data. What we can give you is a much better sense of the ranges and conditions of mercury that are out there in fish. And that's a huge step forward, because we didn't have that because of these two problems. So, what's the status of fish across the St. Louis River watershed? I'm going to give you 3 examples. I'm not going to show you 13 fish and 13 plots. I'm going to show you a few important species that tell us something. This is the first one I thought was really interesting. These are channel catfish. For those who don't have a deep affection for channel catfish, I happen to. I think they're pretty neat animals. Their habitat are main stem rivers. They like high flow environments. They like highly oxygenated waters. They tend to stay down in those channels. We tend to find them in the lower part of the St. Louis River in channel environments, hence the name. They're a nice fish because we caught them at 3 locations in good numbers. Up just north of Cloquet, around Cloquet, and down in the estuary. So we have a nice comparison there of these 3 different main stem habitats with channel cat. So what I'm plotting you here is the size of the fish against the amount of mercury. Now here's our levels. 0.2 becomes a health hazard. 1, if you're pregnant, you cannot eat. 3, which will be way up there, is if you're a healthy adult and not pregnant, you cannot eat. So our highest level here is 0.8. So all these fish are in fish consumption advisory zone. Why are we plotting them as a function of length? Because as the fish get bigger and grow, they bioaccumulate more mercury. So when we compare 2 fish, the size of the fish always makes that comparison difficult. We can solve that problem by looking at the data where the fish are now plotted in relation to their length. So what you're looking for are systematic differences in these fish. And so when you look at it, yeah, it looks like the Scanlon Dam are a little higher. They have slightly more mercury in size. And fish down in the estuary, St. Louis Bay, are a little lower, and similar to those fish caught near Pine Island. And, there are obvious big differences. Right? The data points are often overlapping. So, not a lot of difference in channel cat. Minor differences spatially, pretty similar across the river, and in no level appropriate for consumption advisory. These are yellow perch. This is an odd collection. You'll notice it because you have 2 very odd clusters of data. We got fish, yellow perch, in 2 collections in good numbers from 2 different areas, way at the top of the river and then down to the estuary. It's sort of an interesting contrast. Every fish that was caught in the upper part of the river was a 1-year-old. They're all 3 inches long. Apparently, I don't know, we need better fishermen up there. We need to bring back a real perch. Down in the estuary, we did a little better job on the range, up to something you might want to take home and eat. Look at the range. Many of them are below that 0.2 level. These would still be above 0.05, so it would still be a consumption advisory for pregnant women, but a healthy adult, no consumption advisory. And then the biggest perch would be under consumption advisory. But notice that up here, in the upper watershed, a few of these fish have pretty high mercury given their very small size. So higher, these fish at 5 inches already have levels of mercury. I mean, that's obviously one data point. We'd love more data. But at least that fish already has mercury as high as fish that are twice as big in the lower system.
So the lakes up near Skibo are really organic in nature, right?
And I assume that's right near the sawmill pond, the old sawmill pond, which is really organic in nature. Yeah.
Question, are the yellow perch, they're piscivorous fish? And
That's a
versus the catfish, what do they feed on?
Yeah, that's a great question. So catfish, big catfish are piscivorous. But they have really omnivorous diet. I mean, they'll eat crayfish, they'll eat big stuff down the bottom of the river. Yellow perch are interesting. Yellow perch at small sizes tend to be eating mostly invertebrates, big bugs, worms, that kind of thing. But at certain sizes, and certainly it's just been observed in certain populations above 9 or 10 inches, they actually will become piscivorous. So they're big enough. And that, to some degree, may explain some of this even, what seems to be even a faster rate of elevation. Could be a switch in the diet. Because if they switch from being off the trophic level, they would start to accumulate mercury even faster. That's a good observation. Yeah. So that's yellow perch. The last one is northern pike. Northern pike look kind of like a shotgun blast on a data chart. This is, again, the size, up to 40 inches. We've caught more record pike in our lives, but that's what we got. This is the mercury level. Here's that 0.2 level. Here's consumption advisory. We've got a few below in that 20-inch range, but the vast majority obviously here, and a few are not consumable by if you were about to have a child when you're pregnant. So the other thing is tremendous diversity. I mean, just huge range of mercury relationship here. It's a lot for the same size, lots of different levels of mercury, and for the same level of mercury, lots of different sizes. But by and large, notice that we caught them in Boulder Lake, the St. Louis River Estuary, West River Reservoir. West River Reservoir is up here. And you can notice that West River Reservoir certainly looks lower than the other two sites. We don't seem to see much bioaccumulation with length, but it looks like Boulder Lake and the St. Louis River Estuary are pretty similar.
What's the percentage of error in measuring the fish?
In the individual fish?
And
Yeah.
how many different people are measuring the fish?
Good question. So when you're so you're talking about the mercury in the fish, not the size?
Yes.
Yeah. So the question is how much error is there in the measurement of the mercury in the fish? The measurement is actually extremely precise. The way it's done is we actually take an entire fillet of the fish, we descale it, we keep the skin on. That entire fillet is homogenized. So it's very thoroughly mixed. We take a representative subsample out of that. That's what gets measured in the analytical facility. This is done by the Minnesota Ag Lab. They have extremely good controls. They run blanks. They run standards. They do a very nice job. The error is less than 5%. And levels that are subpar per million.
It's just measured in one place.
Yeah, they're all measured at the Minnesota Ag Lab. Yep. But the entire, that's a good point. The entire, just so you're aware, pretty much all the state monitoring is a contract with the Ag Lab. Yep. So, that's our own one. And then the last one, which is sort of the poster child for this conversation, so I know we'd end up on this one. Walleye. This is, we got good numbers of walleye from two different places, from the Whiteface Reservoir and down in the estuary. And there's two interesting features about this data, so we're going to spend some time on it. Again, we have length, about 8 inches out to about 25 inches in this data set. Here's that 0.2 level, so consumption advisory kicks in here. A few right at that level, where no consumption would be advised for pregnant women. And this was remarkable when we saw this data plot. So, here's the reservoir, and then here's this tremendous variability of these walleye. Tremendous variability. So, walleye at 0.2, right at that consumption advisory, could be anywhere between 8 and 20 inches. We said, what the heck's going on? That doesn't make any sense. So that merits some extra exploration for sure. And it was just obviously in the St. Louis River estuary population, there was something different about them that needed exploration to understand why it is they have such a diverse pattern of mercury. And when you look at it, it's almost like there's two different animals here. So that's interesting. So we wanted to dive in. Rich. All right. I'm going to, so my deep love is for, as Rich said, was from a scary word called eco-geochemical tracers. I will not talk about eco-geochemical. You're going to have to trust me on this one. One of the nice things about eco-geochemical tracers in fish is they tell us a lot about where fish come from. We sort of think of it as fish CSI. These are compounds in the environment that get taken up by the fish, and they get taken up in the place where it lives. And it gets retained in some way in the fish tissue or in the fish bone. And we can actually measure that and understand how the fish is spending its time. And so we thought we'd do that kind of technique with these walleye. And I won't one of the neat things, the pattern we saw, was that the tracer told us that there was something going on. It actually told us that a lot of these fish were spending lots of time in Lake Superior. A lot of these fish had a tracer signal that was clearly indicating they were feeding most of their diet from Lake Superior. And we thought, that's a clue. Maybe that's what explains this. So the first thing I did is I took that tracer data and I made an equation, and I'm going to spare you the math, and we estimated how much of the diet came from Lake Superior based on the amount of tracer in the fish tissue. And it ranges, so it ranges from 0 to 1, where 1 is all of its diet from Lake Superior, basically 100% if you will, down to 0, all of its, so this would be a fish that takes all its diet from St. Louis River estuary. And then we plotted that as a function of mercury. And sure enough, there was a pattern. That the more time the fish spent in the river, feeding there, the higher its mercury, the more time it spent in Lake Superior, the lower its mercury. So we thought that's an interesting pattern. But we know length's an issue too. So I went back to my length plot, and then I took these fish, this cluster, that was getting half their diet from Lake Superior, and I colored them blue. And then I just colored those green. And so what do I see? We plotted that out. So here's your blue. So here's your Lake Superior oriented fish. Here's your river residents. And you can see that the outside band of this scatter plot is entirely filled by those blue. What that means is that these fish routinely have, over an entire size range, have lower mercury at size that they're feeding out in Lake Superior. And this was really compelling evidence. What we were seeing was a migration phenomenon. And that our walleye that were lake residents were coming in, they migrate out to the lake at all kinds of different sizes, coming back in. We're catching them in the St. Louis River, and the contaminant follows that switch in habitat. One last plot. So I made a fancy plot of that. I just want to show you, this is actually important enough I want to include it, even though it's kind of hard to look at. This is the proportion of the diet from the lake, from 1 down to 0. So here's all our lake residents along this part of the plot. Here's all our fish in the river on this side of the plot. And here's their size. So we're going to take these two variables, include them together. And so these are small fish and big fish. So if you follow these two lines up, and here's your mercury, a fish that grows in the river its entire life, by the time it hits about 30 inches, is about 0.6 ppm. But a walleye that spends its entire life in Lake Superior is about 0.6 ppm. But a walleye spends its entire life in the river feeding. By the time it gets to the same size, about 30 inches, it has twice as high mercury levels, 1.2 ppm. So where it's feeding has a huge impact, ultimately, on the size of that large walleye and its mercury level. And this simple explanation of our data explained almost 90% of what was going on in the data. It was remarkably explanatory in accounting for the data pattern we saw. So, migration is a big piece to this system, which makes sense. It's an open river, we know fish come and go. And Lake Superior is a much less contaminated place than the river. But we can pick that up with these tracers. So here's the impact to you. I mean, this is where the rubber meets the road. This is based on, if you take the Minnesota Department of Health and walk it against that plot I just showed you, here's what you come up with. This is the consumption advisory. Yellow means more than one, only eat once per week. Orange means once per month. Red means do not eat. The top table is for adults, healthy adults. The bottom table is for high risk. So those would be children, pregnant women. This is your river resident, high exposure. This is your lake migrant, low exposure. And so you can see that these large walleye, even these large walleye, would fall under a lower restriction, a much lower restriction, if it was spending its life in Lake Superior than if it was spending its time in the river. And that same walleye, spending its life in the river, would be not edible for a pregnant woman. So that's the on-the-ground impact. You can see that where it spends its life actually has a major impact on potential exposure to the mercury.
Most of the walleyes don't come with home addresses.
So our next symposium, as called it, will be turning fishermen into experts in ecogeochemical tracers. It's really tough. In fact, and I know most of you know this, so I'll just get this out of the open. So there's certainly this folklore on this fishery that the silver belly, yellow belly hypothesis. Many of you hear it on your head. We are very, very interested in this. So last year we went out with DNR and went onto the spawning grounds. And we took pictures, about 350 fish caught in the nets that were being pulled as part of the normal walleye sampling for the usual assessment. And we took their pictures, and about 150 of those we took a fin clip. And in that fin clip was that ecogeochemical, that tag, that tracer information. And we analyzed it all out. And we got a bunch of those yellow bellies and looked at them. We only got a few silver bellies and looked at them. And there was nothing going on. In fact, the only one that all the DNR folks from Wisconsin and Minnesota and the EPA people looking over this one poor fish that all agree this thing is clearly a silver belly. We are also thoroughly convinced we finally caught what we all believe is a silver belly. We ran that fin clip and it was a river resident. So I got nothing on the home address. It'd be nice. We did some other morphology work on those fish, looking at some different aspects. I think the silver yellow belly is actually more about spawning stage at this point, based on some of the data we have. But we can look. If people get excited about it, we want to look into it more at this point. But right now, we don't see an obvious way to tell them apart.
I guess the safest thing would be to eat a fish that came out of the lake versus the river.
So there'd be one, right? So if it's in the lake, it's probably spending more time in the lake. The other piece, actually, when we looked on the spawning run, one of the interesting things is that when you're in the spawning run, most of the fish we're catching are lake migrants. When you think about it, Lake Superior is a vast place relative to the river. And so it's just bigger habitat, more fish. So honestly, just by sheer odds alone, you'd be best off fishing the spawning run because most of these fish are lake run. Just by odds, because it's just a much bigger part of the population, much more habitat.
How's that fit with the sampling on the North Shore? The sampling of the babies with high levels of mercury?
That's a great, so the question is, can we link that up to the Minnesota Department of Health study? I had actually a conversation with Pat McCann about this. The simple answer is it's not likely to be possible at this time. And the reason why is because in those studies, they weren't asking people about where they were eating fish from. And so we've actually been trying to start a dialogue over the past year and get some support to do more detailed creel studies on the river, asking people about are you to take that fish home and who might you feed it to? Are you going to share it with your family? Who's in your family? So we're hoping to get some support in the near future in partnership with the DNR and Department of Health to see if we can get some resources to do that. MDH is doing a really nice job getting better numbers on exposure, but we're having a hard time understanding the relationship between the places that have high mercury and where people are actually getting their fish from. That's a link we need to do a better job of. Yeah.
Is there a conflict between the amount of mercury in a fish and what we're told to do when we fish, which is to keep the big ones and throw back the small ones? You keep the big ones and throw them away?
Well, so I think if you go to the MDH website, they're really encouraging you to keep the small ones now. But you're right. So in some cases, we have slot limits that want you to keep some of the big ones and leave a protected slot out. Or the slot is in a kind of a big size. And actually, one of the things that we have not done a good job in this state, especially when we have those slots and we're trying to really encourage people to take certain fish certain size, we've almost never walked that carefully against local health data to see if that's all jives together. It's a good point. It's an awareness I'm hoping is growing after this discussion. But those are two different spheres. Literally, you have one group out here setting fish rags, trying to establish population goals for fishery. Another group here very worried about what contaminant levels are. That's tough. Yeah.
Is anybody studying the other creatures that feed fish besides humans? Well, mink and those kinds of mammals, but also there's a high population of birds that are eating fish.
Yeah.
Well, the gulls don't
Yep.
catch their own very much.
So there's a swallow study going on right now. That's what I'm going. They just have swallow boxes along the river. Now they're eating the insects emerging out of the sediment, but that's a source. So that's been going on. There's a growing interest. I think this will happen next year or two, maybe the next year, in conducting a mercury study on the terns on Interstate Island to look at mercury in the feathers and so forth. Birds are actually really hard because they actually can get rid of a lot of it. They can actually shed the mercury out when they molt. So it's actually really, the hard thing about fish, they don't have a similar mechanism to just flush the mercury. So they bioaccumulate steadily. Birds do this with their mercury levels in their blood and feathers because they can just dump it all. And then, and particularly problematic, and for birds it's usually a concern, is they can actually dump a lot of that mercury into the egg when they're on the nest. So if you have a seabird, an aquatic-oriented bird on the nest in mercury-high waters, those eggs tend to have fairly high mercury because that's what her body's doing. It's flushing it into the egg when the egg's developing. So I think there's some awareness now, and I think there can be support for term study in the future. That's an obvious candidate because they're resident and they're nesting and they're a species of conservation concern. Yeah.
I have a question about land cover. You mentioned that the deciduous trees, when they drop their leaves, that's a mechanism for the mercury to get into the water. Is that as much of an issue when you're dealing with more conifers on the
That's a great
landscape
question.
or not?
Yeah, so this is interesting. So the question is whether or not conifers or deciduous trees are better at enhancing that deposition. And quite a few studies have shown that actually the conifers are better at it. So in conifer dominant watersheds, you actually have more efficient transfer of wet and dry deposition than you do in deciduous forests. There's various hypotheses why. I don't want to get into the science speculation, but it's been measured and replicated well.
Does that tend to accumulate until the forest is disturbed and then that's when it's released? Is that, or does it gradually release?
Well, some of it is the litter itself being mobilized and moved, but some of it is the breakdown of litter and its constituent components that have been broken down, mobilizing. So, yeah, there's lots of interest in this has been shown in a few studies, mixed results, whether or not recently cut down forests have near water, like a pond, where there's more mercury in the animals under forestry management. And there has been some attention to how forestry management practice may interact with mercury cycling. So it's a research topic of interest.
This mercury presence is from airborne mercury, from atmospheric haze that could be either from China or from western coal-fired power plants.
Yeah.
Primarily where it's coming from, right?
Those are major sources, absolutely. Yeah. I mean, there are a few that we just talked about one. WSSD, inevitably, people put products down the sink that has some mercury in them. And that's, or they come off roadways, and because we have a combined system, and, together. So that, there are some of those.
Might be a little out of your realm, but I bet you know the answer. How contaminated are the fish in Lake Superior and Lake Superior itself, being as big as it is? Is it washing out as
Yeah, that's a
clean,
great
or is
question.
it dirty up to 2 hours?
Yep. I have a Great Lakes slide at the end, but I'll give you the quick version of that. They're going down, it's getting better. They are very hard to compare, I'll say this, they're very hard to compare because the good mercury data we have for the Great Lakes come from 2 locations we sample routinely off Keweenaw Bay and the Apasa Islands, and we focus on lean lake trout, which is a really good way to structure a monitoring program to check for changes over time. You pick the same animal at the same size and do it over and over and over again. It's a very good scientific method for doing that. What is very hard is to tell you how a walleye swimming in Lake Superior in the western arm compares to a lean lake trout feeding an offshore food web off the Apasa Islands. And we don't have a lot of food web, very few food web studies, in fact, to make those connections. So in that respect, I think this is illustrative that indeed the walleye spending a lot more time in Lake Superior have about half the mercury levels of those in the river. I'll jump ahead here. I think that's a good jumping off point to kind of bring this back out a little bit. Oh, one more piece of the clue. Yeah, so go ahead. So we were very interested in the spatial pattern in the migrant, so I think we pushed it a little further. Go to the next slide. This is a busy slide. I'm going to walk you through it. We'll get the lights down so you can.
Sure.
Just on the front so you can see this one better. So what about the other fish you said? That was the walleye. So here's the yellow perch, the crappie, and the northern. Let me walk through it. This is their size. We have to, again, we have to that size issue when we talk about mercury. This is the level. Now I put it in a log scale, and the reason why I did that, it's hard to read, I know, but it makes these lines nice and straight, because otherwise they curve up and they're hard to look at. So this is, visually, this is a much more way to interpret the data. Let me focus on yellow perch first. Yellow perch are in blue. Those are all the sites from across the river. We sampled all over. This is the blue. And it turns out that one line fit all the yellow perch. They were, no matter where you caught them, they basically had the same mercury size. Black crappie were a really different story. We could actually, black crappie caught out in Eloise Bay had much lower mercury at size than those caught down in Superior Bay and St. Louis Bay. Here, Superior Bay, St. Louis Bay. And they were much lower than fish caught up here in Spirit Lake and up toward Fond du Lac Dam. And that was, those were real differences in the data based on statistics. Well, that was interesting. So more mercury upstream, less mercury at size downstream. Well, so we looked at Northern Pike. Northern Pike actually showed the same feature. It's a little messier. Northern Pike just needed messier data, but it is, except for this outlier, it fit very nicely that the same thing was true. Fish caught in Superior Bay had lower mercury at size than the other fish caught. Fish closer to that lake had lower mercury. So why would that be? The simple answer is I have hypotheses. I don't have exact answers. And there's a reason why that is. Lake Superior, let's come back to the bidirectional flow. Lake Superior drives water into the two entries multiple times a day, forcing water in. In fact, we measured it. On average, the water at Black Neck Bridge over the years only about 75% river water, 25% lake water. It's a huge influence. And it basically dilutes that water. That river water gets diluted before it even gets out to the lake. It gets diluted in Superior Bay. So there's one piece of it. So maybe mercury's lower because Lake Superior, for whatever reason, has less mercury, and maybe that's going on. Hypothesis 1. Hypothesis 2, when it comes in, there's almost no dissolved organic carbon in Lake Superior. Very, very low. That's why it's so clear. You can see forever. There's very little mercury in that DOC. And so the DOC levels down here, those molecules carrying mercury are much lower because it's diluted out. So maybe it's just that there's, we know that when DOC drops, there's less mercury. We know it drops down there. Hypothesis number 2. Also note that when you get down here, there are almost no fringing wetlands. It's pretty much entirely an armored shoreline. So there's not much sinks for mercury to accumulate in those wetlands as the sediments build up. And there's not a lot of places where methylation can occur because we don't have these big organic rich wetlands. We have sandy sediments on the dunes and a lot of armored shoreline. So maybe it's just a wetland feature. The wetland thing's interesting because if the wetland thing was true, then you'd think you'd see a tick up again in Eloise Bay because it's certainly no lack of wetlands in Eloise Bay. But then you get the crappie. Eloise Bay has a lot of Lake Superior water in it. It's about half Lake Superior water. All that water runs out of it. It keeps Eloise Bay surprisingly cold. Fishermen know this. It keeps them cold in the ocean system for a lot of the year because all that water's flushing in. So the short answer is I don't know, but it's interesting. And one of the problems we have with these kinds of analyses is that all those things are happening simultaneously. So it would take us quite a bit of time to tease all those out. We could do it. But that's where we are with this data set. So spatial patterns, too. I mean, it's not just simple. It's not just an estuary. There are different parts of the estuary, and they have different mercury levels in them. And so where you measured it and monitored it would matter, because they have different mercury levels. Last bit here on estuary fishes. Back to the geochemical tracer. I don't want to belabor this. This is the level of mercury in the fish as a function of the amount of 15N. That's the heavy isotope of nitrogen relative to the normal isotope of nitrogen, 14N. And I don't want to belabor this. It turns out that this increase happens because fish in the food web, as fish feed higher, they actually accumulate 15N. They don't just accumulate contaminants, they also accumulate 15N. It's a real nice independent marker of trophic position. So we can use that data point to plot a mean and variation around these fish. Black crappie, yellow perch, northern pike, walleye in the upper estuary as a function of mercury. And what you can see here is that as they go up the food web, they go up in mercury. We did the same thing in the lower estuary because remember they were slightly different. Mercury levels, well, maybe slightly different. Let's just be careful, take a look. Black crappie, yellow perch, northern things come out of this plot. One is that they're pretty much, both food webs are fairly similar. They're oriented the same. The fish are in the same relative places. That increase is about the same as a function of it. That's interesting. And the other piece that really blew me away was that the northern pike are consistently lower than walleye and the food web. And we kept wondering why are northern pike lower than walleye. I thought that was kind of weird. I was expecting northern pike actually, they're not quite as long-lived, but they're a long-lived animal, and when they're big and piscivorous. Well, they kept coming up lower in Mercury, and this is why, because they're actually getting lower on the food web. So there you go. Next slide. Last slide on the estuary. We'll back out a second. This is the time series data that I've been able to put together so far. This is my big plot. These data are not exhaustive. Bruce wants to, on our big data mining exercise, we're going to get every shred of data on Mercury on these fish we possibly can. This is what I've assembled for the lower estuary to date. Green is back pre-1985. I think those data go back to 1979. The squares are 1991. The triangles are 1987. The blue is what we did in 2013. Size here, mercury level here. And the point is that there's not an obvious decline over time. All these points are on top of each other. So it's not obvious, at least from this data set, again, not exhaustive, there's my big caveat, but what we've looked at so far, we don't see an obvious trend of mercury declining over time since the late 70s, early 80s in this system. At least in walleye. But no, walleye are really kind of not a great monitoring target because of this huge variability in them. So they're not very sensitive, because we're catching some years, we're getting some migrants from the river. It's what we've been monitoring most consistently over the years, but it turns out it was not a great choice, because in any given year, you can get a lot of noise in the data, because you get some mix of rivers and some mix of lake runs. And you don't know, going back in the time series, which one are which, because we weren't doing equal geochemical tracing back then. So I can't tell you, going back to this data set, and you could say, oh, maybe that one's a lake and that's a river. Well, we don't know. Maybe that's a lake, maybe that's a river. We don't know. But it's not a great monitoring target, as it turns out, for reconstructing time series because of this migratory behavior. Next slide. Comparison to other waters. So mercury is high in St. Louis River fish. Pretty much all the fish we look at require some local consumption advisory. This is the map of fish. Every time you see a red dot, that's a fish over 0.3 ppm. That's the water body with the consumption advisory. This is for walleye. This is for northern pike. We have a regional problem. We have a lot of water bodies that are clocking in similar levels of mercury. And we have a lot in Minnesota. A lot concentrated here on the border. Here's the St. Louis River, but also outstate Minnesota. And that's true for both walleye and pike. So it's not just us. This is a geographically widespread problem. It extends through Ontario into the UP.
So is that slide showing kind of the same thing, that the walleye actually are having a worse problem than northern pike?
Well, so, not necessarily, because walleyes, on average, tend to grow, populations tend to be quite a bit older than northern populations.
Okay.
So, given that they're out there longer, they more tend to bioaccumulate. It's not obvious that would be the thing to do. But the trophic global phenomena we saw extends to other water bodies. There was some good news. Let me finish. That was all kind of depressing. So let me finish on a couple of notes of some good things going on in the region. This is walleye across the Great Lakes. This is work done, summer work that Bruce Munson did a couple of years ago. And this is largemouth bass. And this is a complicated model. They got data from every single point where you see a black dot here. So these were huge data sets. So very regionally comprehensive. The kind of data that's really useful for complex environmental phenomena like mercury. And in both cases, the walleye, largemouth bass, you can see that since the 1970s, mercury in parts per million, 0.4 here, 0.35 there, a clear decline in both species. And some other species too, it's all the same phenomena. Regionally, mercury is on the way down. This makes sense, and it corresponds, of course, with the period when the Clean Air Act was enacted, when we started doing some very specific things about changing our air pollution. We started being much more conscious of what was coming out of smokestacks. Importantly, we started reducing sulfuric acid inputs, which were a big regional driver, and that's all good. So it's going down. The problem is getting better on a regional basis. Where are we going? In 2014, UMD and Gustavus Adolphus partnered and started doing some nice work looking at mercury and potential AOC restoration areas. We're hoping to see data out of that very shortly. That's the first time we've had a really good look, in-depth, site-specific detail on mercury values in places in the estuary. Concentrated effort, very careful. That's a great step forward. 2015, it looks like what's going to happen. I know not everything's signed here, but everything's pointing this way. There's going to be some kind of sediment flux study. There's a lot of interest in what happens when we start moving mud, creating wetlands, what that means for mercury methylation. And I think there's going to be some concerted effort to get a study underway. This has, for those who might recognize, this does have a relationship to some degree to the TMDL process. And then it looks like Wisconsin DNR on their own is going to initiate a food web study. I'm hoping this comes to fruition. They'll probably work with us and some other partners and really do a better job focusing on lots of different, not just fish, but other parts of the food web to tease out some of these problems. Hopefully we can do it spatially and figure out what parts of the system are really problematic. Folks involved in this problem got together. This is what we proposed for the AOC, not the TMDL, for the AOC, the Area of Concern process, what we need to do. Top on our list was to get together all the data we have. It's been collected for years. It's not together in one place. 2 different states, different people, different times. We're going to get it together. We need to find a way to standardize contaminant data in fish tissue. It's almost all from ice, thankfully, based on size or age. And you've seen the problems while we have to do that. We'd like to start developing and deploying local scale biotic indicators. So these are bugs or little fish that can tell us what's going on at a site, which would give us better ability to actually tell you the mercury is high over there, not just in the upper estuary, but really start to bring that spatial scale down and be more specific about advice to the community about where the work needs to occur, where are all the problem spots. And to that end, we'd like to do something more comprehensive, actually a further more comprehensive study, where we actually do a specific, much more explicit job contrasting the stream habitat, the main stem, the reservoirs, and the estuary, because they'll have very different environmental features. And getting at some of the different food webs in them, we start to tease apart some of the issues there. Because it may be very well that it's some of these food web pieces that are driving some of the patterns, not so much on the contaminant side, but more on the biology side. We need to know that if it is, so we can weigh those different aspects of the problem.
One more slide.
I think that's my last slide. No? That's my last slide. So just to round this out, we can do Q&A. I know you're all tired. That was long. Thank you for working through that with me. So mercury fish has declined across the region. It's done so in tandem with reductions in atmospheric pollution. That's a good sign. Total suspended solids have declined in the St. Louis River. We know higher TSS is generally associated with higher mercury levels. That's a good sign for the river. It's improving health, has some positive implications. Not wholly positive, but some positive implications for mercury fish. Nevertheless, mercury levels in St. Louis River fish are high compared to other Minnesota waterways. And observed levels are similar to the other high mercury waterways in the region. The geographic setting and ecological properties of the St. Louis River make it vulnerable to mercury bioaccumulation. The forests, the wetlands, the connectivity, the low productivity, the reservoirs, the changing water levels. We know that fish, we now know, we didn't know this is where we made some progress, fish vary mercury concentration across the watershed. We get different levels at different places. They even vary within the estuary. And what that tells us is that we do have to do more work on some of these local scale environmental factors. And 2 big ones we talked about this evening, one being movement, habitat positioning within the estuary, habitat with respect to Lake Superior. The other one being trophic position, for example, why are walleye higher than northern pike systematically? So that's where we are at this point. What else to say?
Question for you. I would assume the St. Louis River estuary is probably one of the worst case situations in Minnesota as far as exotic species go, exotic problems.
That's probably true. That's probably true.
At least
We are among the most invaded waterways.
Right, the
That's a fair
exotics,
statement.
and exotics are getting a lot of attention in Minnesota. From your study of the St. Louis River estuary, which are you more concerned about, exotics or contaminants? Which should we be spending more effort on?
I think contaminants is the right for now. honestly. I mean, and I say that in the sense that the contaminant levels we have are not a threat, do not present, except for a few cases, there are a few sediment areas that are a threat to fish and wildlife. But by and large, the sediment levels that we are left with today do not have a direct threat on fish and wildlife health. They have a huge impact, however, for human health. And if we want to restore this community's relationship to the river, this is a very important piece. The invasive species that we have, the only two, we have a lot of non-native species. Most of them occupy small and rarefied roles within the estuary. Only a few have we identified as having big players. The two are Ron Goby and Roth, and at least for now they're in check. And for whatever reason, the system is changing and they're declining. I'm playing a smaller and smaller ecological role all day. And so that would be my argument, is from a conservation standpoint and from a science standpoint. I mean, this is, in some ways, this is tougher too.
When you look at protein sources for people, what's the impact of this for food for humans?
I mean, it's tremendous, in the sense that what we've done is we've taken a local sustainable food source and made it not a food source. So then what we have to do is find other sources of, if we want to eat fish, we go find other fish to eat, or we alter our behaviors. And we know from creel surveys on the St. Louis River, there's some data, but we know that it also behaves, it changes people's fishing behavior. They behave differently. When people know there's a consumption advisory, they fish it less. So it has lots of impacts in that sense. On the flip side, we can. People who have options will utilize them. And so if you have the resources to go buy a different fish that has less mercury in it, you can do so. You can go to the supermarket and you can read up on it, and you can get one of those little fish watch cards, and you can make a choice. But there's a lot of the population out there that doesn't have that. They're fishing subsistence. They're doing it by choice, or they're doing it because this is what they eat. And they don't have that kind of option. There's a reason why there's lots of people focused on this. And we, public, as a community, we agree this is a huge problem. I mean, that's why EPA and so many PCA and MDH are so invested in this problem. It's because everyone agrees this is a tremendous amount of impact that needs to improve.
You mentioned that there was a local lab that was doing tissue work for you.
The ag lab, that's the state ag lab.
Ag lab?
Ag, agriculture.
Oh, ag.
Yeah. So Department of Ag has their own Mercury lab, and so they state contracts with that.
Question in the back there.
Yeah.
Now, one of the things I noticed that a lot of short fishermen and stuff wasn't okay. There's a lot of guys who don't believe in this and are taking, I see them all the time, and they're taking these fish. I mean, they're eating all sorts of fish every week. And I know that, it's out there, we all know.
Yeah.
But I just really wonder about those guys.
It's true. I mean, there are certainly people who do not regard consumption advisories, and we post them here, we post them at docks. Folks at the DNR and Department of Health work very closely to make these very legible cards and pamphlets you can download.
You saying you wouldn't eat fish out like St. Louis River?
I would not eat fish.
Pardon?
That I would not?
Yeah.
I would eat some fish. I mean, there are some fish. I mean, you could eat an 8-inch yellow. I would eat an 8-inch yellow perch on a spirited bag. Yeah, that fish is. There is based on our data. There's no consumption advisory. Would I eat a walleye out of St. Louis River? No, I would not.
You wouldn't?
I would not.
And
Our family, we do a lake carrying share, so I love fish, and we made a choice to sort of focus where we know there's less pollution in our eating behavior. Yeah.
There's a great irony there, because the river is now using walleye have come back, and therefore the river ecology and water quality have improved. I mean, we hear that all the time.
Exactly. It is. I mean, the fish, the community fish community is thriving. There's tremendous biodiversity. The walleye population is doing quite well. But we have this other problem.
Question probably isn't a good one to ask right at the end here, but I think you'll live in this community with mining, and it's gonna be, somebody's gonna be here for years to come. What is the threat, do you think there would be, is there a threat from sulfide mining on the lower reaches of, or the estuary down this level?
Can we walk that one carefully, because that's, there's like 5 mines, right? PCA has, Mike Byrne at PCA has done some very nice studies trying to understand the existing systems. First, let's be careful on that. We need to separate 2 worlds. The one in which we have the current taconite mining, and it's contributing sulfate to the earth. We know that. And so, I mean, there's been a series of systematic studies conducted by
Yeah,
Mike
the other
Byrne,
half of
PCA.
my question was, I was gonna ask, how much has
Right.
mining as it has existed up there?
Yeah, and that's really tough one. We know, so what we know is that the mines are definitely contributing sulfate to the river. We know that. What's been much more difficult to figure out is whether or not that sulfate is actually stimulating methylmercury. The work that's been done suggests that the river in its current condition, as is under the current influence and the legacy of pollution, is not very responsive. But in a sense, that's not what you want to know, right? What you really want to know is in a world where that legacy could be remediated for, how would the system respond? And that's an experiment we cannot conduct in many ways. It's a best of thought experiment. We don't have the data on the river's condition before that impact was there. So I think all of us involved in this issue are really hesitant to say, on the one hand, that given that today we know that the sulfate being added isn't a big generator for methylmercury, be hesitant to say in some future condition it couldn't be implicated, because sulfuric acid and sulfate have an impact on methylation. But again, we also don't know that in the past, methylation, some level, didn't go on without the mines. It's a really, there's no good way to answer that. Nate Johnson did some real nice experiments. They've been published now. They added sulfate to some sediments. The sulfide bacteria, the sulfide-reducing bacteria responded positively, but they didn't get any more methylmercury out of them. They're using estrogen sediments. But again, there's that caveat. It's existing waters, existing sediment, and that's not really exactly the thing we want to know. So it's a tough question.
One more question.
Yeah.
This guy back here.
Follow up on that do you have any information on what the mercury concentrations would have been pre-industrial revolution at all?
There is a net, so yeah, essentially yes. And we know what the natural contribution is. And we know that they would have been about 30% of what they are today. So we do, I mean, we know that. And we know that we have radically altered the geologic cycling of Mercury. We've extracted, globally we've extracted a tremendous amount of geologic reserves and put them into the atmosphere. And we've done that through processes like burning fossil fuels and mining, and I could go on and on for a while on the sources. But it is a clear massive perturbation to the cycle of Mercury.
Do we know about the mercury levels in fish, prior to industrialization?
That we do not. And there's no... Agassiz did leave us a nice collection.
Making good public policy is really easy.
Yeah, it would have been nice. We don't. There's coring work to try to understand. That tells you a little bit, not a lot, because ultimately, you may have got this, a lot of times there's a huge disconnect between how much mercury is say, in the sediment and what's actually in the fish.
Right.
They're often very rarely related. They are almost never related. So even getting coring data will tell you what you already knew, which is that in pre-industrial times, mercury was lower. We know that, but it doesn't tell you about what's going on with fish.
As we wrap up, there's 2 things I want to do. First, I want to thank Joel, but before we do that,